And welcome to our first seminar of our fall series for the Pacific Southwest Center of Excellence in Vector-Borne Diseases. And today we have the pleasure of hearing from two of our key hosts of summer internships of our center. And what we're hoping for this session is, for those of you who haven't hosted an intern, perhaps this might give you some ideas about how these arrangements can work. We're also planning to put out a call in the next week or so for ideas for next summer's internships. And those can take many forms. Many of you in our region have your own resources for hosting interns, and they end up being wonderful experiences when we can couple students from our 2- and 4-year colleges with you, and then ideally get experience that might lead to future employment opportunities or just great experience that gets people excited about work on vector-borne diseases in our region. Today we have two wonderful speakers. First up, we have Dr. Chris Bibbs. He's currently the laboratory director at the Salt Lake City Mosquito Abatement District in Utah. He has a master's in entomology from University of Arizona here in our Pacific Southwest region, also a PhD from the University of Florida, and he also has some experience working in industry before he joined the Salt Lake City district. And his district's long been an important part of our PacVec Center. And last summer, they happened to also host at least one intern working with our center. So Chris is going to tell us about that experience now. Chris, over to you when you're ready.
Sure. Yeah. So if you don't mind, I can do some screen sharing on my side. That way I can control my thing. So to preface a little, so your two talks today kind of come from two different angles of, I guess you could think of like bureaucracy. Our district is independent, so I have a lot of freedom about how I approach the process, as well as like odds and ends, dexterity within the program, how many people I can take, and even the amount of discretion I can allow the individuals to participate in. So what I'm going to be showing you is more along the lines of like a freely navigable program, something where like you don't have like a ton of oversight, but you might have issues with like resources or something like that. Whereas Megan's going to be presenting more from like you have a very discrete structure you have to fit into, and how do you navigate that framework, especially on a state level, and still get the job done. So to preface ours, you can see this playing, right? It's like a little video. So this is our facility. We're the Salt Lake City Mosquito Abatement District. We're a 100-year-old abatement, the third oldest in the US. So don't let this like intimidate you. But the reason I'm showing you this is to give you an idea that we essentially have like university style facilities that we can have these interns in. So when I say that I'm the laboratory director, I have multiple spaces that I work in. We also have a lot of ability to accommodate people, including living space, detached and attached laboratory spaces, and the ability to kind of partition people around and split them up. So I am very fortunate to have a very significant amount of infrastructure at my disposal. So I'm giving you this information with that caveat that, you know, we're a very well-established district. We have a long history with this. And so what comes with that may be what for you guys are seeing it is like, this is the long view. Like after a long time of doing this, you might have something like this. So don't let this like intimidate you, because even for us, we started with, you know, the occasional one intern. For an idea of how my operating budget works, and the reason I do this is because I know all of you guys have different budget situations. You maybe have your individual lab, or you're operating off a grant budget, or maybe you do have like a partial like state funding or something like that. My laboratory operating costs ends up being anywhere from 100 to 200,000 a year, and that is everything. That's the supervisory cost, the staffing cost, the equipment cost, the annual budgets. So that includes me in there, as well as, you know, all the resources that people use. And the reason I put that down there is some of you have different systems. So if you're in the university, for example, you're probably not going to factor your personal pay into that, but you might for your postdoc or something like that. Or if you are at an independent agency, you might need to consider, you know, if you have a molecular biologist and what what that experience is going to cost you to have them also presiding over interns. So I put that in here just so that you have some context. We have two discrete budgetary breakouts. I have a molecular biology budget and I have a surveillance and biology budget, and these do different things. My surveillance budget is actually to set and collect traps across the service area within our district so that I can monitor and inform for operations. So I actually have a mission that stands separate from the internships themselves. And then the molecular biology budget is in-house PCR testing. We have the option of a state lab, but for us, it's a lot more time efficient and a lot more like control functional to just internalize that process. And so we have a molecular biologist on staff as well as a lab with a budget to be able to process quite a few samples. And so what our staff load kind of looks like is there's me, and I have a secondary supervisor, which can either be a biologist or in this case, a molecular biologist. And then I have a certain amount of allocation for seasonals, as we call them. So these are people that work no more than 3 to 6 months in the year, but that may be during the summer, that may be during the winter. And we actually advertise these positions. And so we tell people these are field work, they're research opportunities, they're real public health applications. You get to work outdoors. It's a Monday through Friday workplace. We can guarantee you 40 hours a week. And right now we start them at $17 an hour. And so whether we get people through PacVec or RapVec or people independently seeking us out to get internship credits with their campus or something, this is kind of a flat offer. So we may get some additional support, say through PacVec or something to support a student. What we usually do is we use that grant money to fund the actual research part rather than the person, in my case. And so the outputs from the combined totality, and I'm talking about the budget and the people, I get to run about 2,600 trap nights per year. This is 40 CO2 trap sites twice a week, plus an additional 22 gravid trap sites once a week. So I get to do differential surveillance. So you could think of this for a lot of you guys that work in academics, this is passive data collection. This is stuff that happens no matter what people's research is on. And although I may use it for operational purposes, this is also like a really significant resource to piggyback on and integrate things like interns and stuff. Because maybe, like in my case, your issue isn't data, it's data analysis. So like I have lots of stuff, but I may not have the time or the bandwidth or the people or even the skill sets to properly ask questions of the data I'm already collecting. From that molecular biology budget, we're looking at 1,000 or more mosquito pools a year. So that's 1,200 PCR reactions to look for things like West Nile, Western equine, SLE, or in this case, this year, we also did some with heartworm. I also keep colonies here. So this is a benefit of me being an independent district is I can do stuff like this and I don't have anybody complaining at me about it. I typically have, 3 to 6 species in colony. Right now, that is primarily Culex tarsalis, Aedes aegypti, Anopheles quadrimaculatus, and Culex pipiens. But I've also periodically had Aedes cearensis, which is a tree hole mosquito. And then in the outreach capacity, we have Sabbathes cyaneus, which is a very beautiful, showy, metallic, kind of feathery-legged mosquito. It is not a vector. It's also tropical, so it drops dead with like a little bit of air movement, so they're a little difficult to take care of. And then I also have Toxorhynchites, which is predatory and beneficial. So I use those in research sometimes, but they're primarily used for education. So within this structure, my interns typically will produce anywhere from 4 to 6 publications per year. This is with assigning them generally 1 project per intern. However, that doesn't mean that the 1 project they get siloed into is the only thing they're doing. So a lot of times they provide extra hands for big projects. So because we're independent, we have a lot of like our own investigations we look at. Maybe we're doing evaluations on a process. Maybe we're looking at efficacy of things like field treatments or something like that. And these exist independently of what I'm doing, but we do help and participate in this. And so these people also will help out with these really big, often 10 to 20 person investigations that we have going on. And then on top of that, and I think this is really an understated benefit of these interns and students, extra skills. These people have education that we don't. And I know that sounds weird because they're coming to us for critical thinking and opportunities and for diversification and skill sets and stuff, but they already have certain education. So like me, I have more of a chemistry background. You ask me anything about plants, even like how to identify a plant, and I'd just be like, that's a tree. That's about what I have for you. I think it's a bush. What's the difference between a bush and a tree? So like if I have a student with a botany background, that is excellent for me because I don't. And so you can kind of think of this as your mileage may vary. However, one of the things that's really nice about this is you often end up with a lot of odds and ends things that you don't have time to look at. So I use traps all the time for collecting mosquitoes. And one of the things that comes up is, well, what trap does the best job? And people, you'd be surprised. People argue about this stuff all the time. Do you use a light? Do you use no light? Do you use heat? Do you use no heat? Do you have an LED light? What wavelength? How warm is your incandescent light? And it's just like, I just want a light on a trap, y'all. Like, I just need to collect mosquitoes. So if I get an intern, this is actually something that I can task them with, and we can look at variable isolation. And in our case, we even have like 3D printers on site and stuff, and we can test prototypes and things like that. So I frequently use my interns to help out with like nuts and bolts type of stuff that I normally would not have the leisure to do. And you may be wondering what the payoff for this. This isn't just tangible physical items that I use in my district. I mean, these are real experiences for these people. So in this case, Zandy Harker, she came out of BYU, and she was more of like a strategic or epi backgrounded public health major. And so this was her only practical applications working in mosquito control. This was her only boots on the ground experience. And this was really significant to her. And it did result in a publication that not only reinforces the knowledge that we gain, but also helps give like some blueprinting for the devices that we use. So there's a lot of long-lasting benefits with that. Some other examples, I mentioned the plants. That actually comes up in my side of the woods all the time. I don't really get to ask just the human-oriented questions. I have to ask the environmental ones as well, whether that's from like environmental toxicology or land use or things like that. And I don't honestly have really good skill sets for those things. So when I have, people, like I said, with a botany background or something, we can do stuff like looking at how mosquitoes interact with the plants that occupy those lands. And we can look at relationships between ground cover and movement in the city and associated risk with that vectorial capacity. So it may seem like these are sidebar things, but they have a lot of direct influence over how I then decide for my surveillance program where operations really should be focusing their effort and stuff. So this has a lot of really good benefits, even if they may seem like a somewhat basic or natural history aligned question. Some other examples, some of you guys may be familiar with like pushback from like social or community or cultural type things. So this is excellent stuff to have interns help with. So like in our case, we have beekeepers and managed bees. We have people that are very reluctant to be in areas that are sprayed with pesticides. And we have people that are, in clinical environments, you could call them the worried well. There's nothing wrong with them, but they're afraid that one day there still will be something that's not wrong, not right with them anymore. And they want to attach, blame to people and stuff. So what I find in our field, I deal with these people all the time. And so sometimes it can be a little exhausting. And you get a little hung up on, I've told you guys 20 times, like, quit being crunchy granola and let me kill your mosquitoes. Would you rather have West Nile and stuff? But like, that's not really a good approach. It's not very PR friendly. It also isn't answering their concerns and stuff. And so in this case, I had an intern that was shared with us with UDAF, and she principally worked with the apiaries inspections unit. And so she conferred her bee expertise to help us look at bee health in association with our just everyday things that we do. And this ended up resulting in some really nice information about, whether or not we actually do present a risk to the managed bees within our area. So we're talking like commercial beekeepers and stuff. We have, the Browning family out here, and we have like the Hawk family and stuff. These are groups that will ship 1,000 or 2,000 colonies out to California during almond season and stuff like that. So like we actually have a basis for a lot of these like managed bees and stuff. So this is really interesting too, because this is a collaboration I would never normally dream of. But because this intern had an external connection that they could kind of like double dip, and use to help with us, that ended up growing into something really nice. I have relationships with lots of other groups. So like in this particular one, this is actually a military project. So I've actually been reached out to by the Naval Entomology Center of Excellence, which is based out of Florida. And they have this medical corps that deals with public health problems. And sometimes that includes zoonotic things like what I work on. And they also are looking for experience in the things. And so you might be surprised, something that may start as like a PacVec program or something, what it does is it gives you visibility. And once you start developing a track record, then more people from other like, sidebar or like tangentially thing, you build this network of potential or potential internship opportunities where others may be interested in working with you as well. So like in this case, this was a aspiring lieutenant that was working on repellents. And so all he was really asking was, do these repellents protect soldiers in the field? But that isn't just a military question. We use repellents all the time. And in fact, in my job, I use repellents. We have really bad mosquito seasons. So even if I'm going to collect traps, I may wear a repellent or something. And so what he was looking at is like some secondary toxicity that may happen when using these repellents. And this too resulted in some publications and stuff. So this is really nice, nice yields that come out of this type of work. Now, some of the other things, one thing that I think is good to consider about particularly like the PacVec involvement and stuff is treating it like a seed start in a way. Not all the yields are going to come from the immediate relationship with that intern. So for example, this project here, we just dubbed it the mosquito attract and kill device. The idea is a passive point source that mosquitoes come to and then die. It's meant to be an option when you have no like physical surfaces that you can treat for mosquitoes, and when you don't know when mosquitoes and stuff are flying through. And so you can see on this little sticky strip here, all the different types of things that it collects. It collects biting midges and mosquitoes and like deer and horse flies and things. And this actually had arisen from interns that had collected the preliminary data. And then we used that preliminary data to apply for a grant with the Western IPM Center, of which all the PacVec numbers are part of Western IPM as well. And so we were able to use that to then build a bigger, more fleshed out project, which we have now been working on for like 2.5 years. And so that's really nice. In my case, I have some subject specific stuff. So I'm more of a toxicologist. And so one of the things that I can do with interns, and this is what I usually do, is I do mad science experiments. So in this case, like I have an engineering friend. The engineering friend is like, I made some fake tissue. Can you use that for something? And I was like, sure. Mosquitoes like to bite things. If it's a fake tissue, then I'm sure mosquitoes would like to bite it. And so one of the things we developed was like a fake nectary, like a plant nectary, and we can load it with whatever we want. And again, this type of stuff is preliminarily funded and supported through these internship programs, but then can grow into larger grant efforts by big societies and things like that. So I think that's a really nice yield as well. And then some of the other things is that these have kind of piggybacked on that growth that comes from things like PacVec. So I mentioned the visibility. I have local universities that are not the UCs that are in my side of town and in my neck of the woods. And when you start kind of getting this notoriety, and in our case, publications and stuff, so you get like little news pieces that say like, oh, Salt Lake City Mosquito Abatement in collaboration with PacVec or something has this, nice project they did. It starts to get attention. And so we've actually been sought out by our local university, in this case, University of Utah, but we've also got Utah State University, which is a land grant, Brigham Young University, and a smattering of like RO2 and RO3 campuses that are all around. And now they've identified that we are a place that students can go. And so they start like kind of sending them to us, encouraging them to find us. And so this is an example with the College of Engineering at the U, and they've done things like building us wind tunnels, making rotator traps for looking at mosquito like deal activity, and even operational stuff, developing devices that we can use for disseminating treatments and stuff like that. And so this became, kind of like self-realizing gains where like these students came to us and they're like, hey, this is cool. I think I'd like to work with you on something. And then we throw an idea out to them and they do it. I've been able to extend this relationship to some other things. So like at the U, I've been able to work with the community engaged learning program that they have. These are university seniors. So these are people in their last semester or two of school, and they're looking for like some job experience. And then we also have this thing called the Science Research Initiative, which is where been putting a lot of extra effort lately, where these are freshmen. So they're people who haven't even declared a major in a majority of cases. And they also are looking for opportunities to see research to decide if they want to be a science major. And if so, what kind of subject specialties do they want to do? And so I've had these students, whether they're freshmen or seniors, also working on like strange tangential things. I mentioned I work in surveillance. A majority of my lab budget goes towards that. I have mosquito species like the Western treehole mosquito. It's a heartworm vector, and it doesn't go to our traps like ever. And so one of the things we were looking at is why. What do they actually like and stuff? And I was, I do chemicals and physiology, so I was just like, oh, maybe there's something in the water. Maybe they're looking at like nitrate versus nitrite content. Maybe there's an ammonia load with some ammonia shock and whatever. And this is one of my freshman interns, and he was like, maybe they just like the color. And I just kind of stared at him, and like I didn't actually have like a good rebuke for it because I was like, all right, sure, we can test that. And so we went through literature to find some stuff that they do like, and we did these kind of visual analyses with it with behavioral assessments. And it turns out these treehole mosquitoes like the color purple. I mean, like literally, we tried this on infusion water, different types of plants, artificially spiking with nitrogen or nutrients. We used amino acids and all kinds of stuff. Nope, color. They like the color purple. Okay. And that is literally something I would have absolutely overlooked if it wasn't for them just having a very simple question that comes from, not taking the information for granted. I assume I already know how this stuff works, right? Interns don't. And so they can ask questions that maybe you just wouldn't have. And that can be really nice as well. Oh yeah, and if you've never seen, heartworms or any kind of filarial worm associated with mosquitoes, it's pretty grody. And even though some of these people are very inexperienced, like in this case, this was a freshman. It was his very first year in school, literally his second semester in school, and he was able to get a publication by working with us. So there's a lot of exchange. This is very much a two-way street. Not all yields have to be stuff like that, though. One of the things is, I mentioned that visibility. We have a women's specific program at the University of Utah called Access, and Access is intentionally geared for women in STEM. And so we were fortunate to have a student from that program. And one of the things she did is she took our work to the campus. And so she was helping us with some surveillance with dark heartworm, because I mentioned, they're kind of difficult to work with and they are a vector. And they have like these mock poster sessions and stuff they do on the campus where other faculty and other students will show up and see, what do these students do? And she brought our work to them. And so vastly improved our visibility to these other groups and has very much increased like how often people, will email us or kind of a ping a bit about like, hey, when do you guys do internships? Is there any program I can apply for? Are there any centers or societies that I could get funding to work with you guys and stuff? And so that ends up kind of snowballing into a whole bunch of stuff. Ever since being affiliated with some of these, I've been able to do a lot of my toxicology work. So I actually had somebody from a diabetes lab of all places, like come and spend some time with us. And he really didn't know why he was working with mosquitoes, but he liked fly fishing and he liked bugs and he worked with diabetes. And so we're like, all right, how about we look at sugar metabolism in mosquitoes? And he's like, all right, I'm game. And we ended up actually finding out that some artificial sweeteners like erythritol and stuff, they actually like impair your glucose digestion, not just like because you don't have glucose to digest, but they're actually like antagonistic to the digestion, and that makes them toxic to things like the mosquitoes. So that's one of those like, wow, who knew? And like, I definitely wouldn't have been able to do that if it weren't for the fact this guy already worked in a diabetes lab. So, I mentioned that we've got all these other like bigger projects and stuff. I don't really want to overstay my welcome. So I just want to kind of recapitulate, what we really get out of doing this stuff. We get visibility with these like voluntary based programs like CEL, which are seniors, or SRI, which are freshmen. It doesn't really matter what it's called, but what you guys should consider it is like, well, what does that look like in your area? Are there any universities or even small colleges that have little programs like this that are looking for involvement? And can you reach out to them and can you point them towards stuff like PacVec or other structures? You may even get some very explicit associations with skill sets like engineering, botany, biochemistry, bioinformatics, and stuff like that. And in our case, we've even had solicits for working directly with graduate students later down the line. So these aren't always early stage trainees. Sometimes these are like a PhD candidate or a PhD student or even a postdoc who asks to work with us because they realize what we can do. And all this kind of comes from us forking over a little bit of budget to have a couple people we support. But it spreads across these other aspects and seed grants and other things. And it becomes a self-building process. I mentioned, looking at like the MACDs or getting stuff from the American Mosquito Control. Those are things you can then use to incentivize more interns to be with you. So I really can't emphasize this enough. Working from a program that's dynamic like mine and that I have a lot of freedom about how I do things, think of this like an exponential growth curve. You're going to start real slow. You're going to have your one intern or something, but that one intern lets you work on something that you wouldn't normally have the time. And then that grows and that builds. And then the next project, maybe you can manage to have 2 interns or 3. And year after year, by the time you get to your 4th, 5th, 6th, 7th year, you end up with this really long, stable relationship with lots of entities that may have all originated from just one opportunity, like PacVec. So anyway, Chris, it's kind of up to you how we want to manage our time about questions. I'm okay to take questions now or later, but I'm also okay to move over to Megan if we want to do that.
Okay, thank you so much, Chris. And we've long appreciated your engagement as well as Salt Lake City Mosquito Abatement, Ari, Greg, and the rest of your team. Certainly the benefits have been mutual, I hope. And we also appreciate all your mad science, as you called it, which also gives people a lot of experience in experimental design and the scientific method as well. I think to keep on time, we'll go ahead and switch over to Megan, and then we'll come back at the end for questions for either Chris or Megan. So make sure if you have them for Chris, just hang on to them a little bit longer. Next up, we have the privilege of having Dr. Megan Saunders presenting to us today. She is currently a senior public health biologist at the California Department of Public Health, which is also an agency with whom we have enjoyed working for a very long time. Dr. Saunders has a Master of Science in Public Health degree from Tulane University. She also did a stint working at the Maine CDC and no doubt working on ticks there, I'm presuming. She has a wealth of mosquito and tick experience, I'll say. She has a PhD in environmental science where she focused on urban mosquito ecology at the University of Maryland. Briefly worked at the Maryland Department of Agriculture working on mosquito surveillance, and then joined us here in California and has, for the most part, been working on ticks since that time. So over to you, Megan, when you're ready.
All right, thank you for that introduction, Chris. Hopefully y'all can see my screen.
Looks great.
Great, thank you so much. Yeah, so my name's Dr. Megan Saunders, and I am a senior public health biologist at CDPH. And we're gonna sort of shift gears a little right now because as Chris mentioned, I do work for the California Department of Public Health, so we have some certain goals and structures that we must adhere to. So I work for the vector-borne disease section, and we do have the mission of protecting the health and well-being of Californians from arthropod and vertebrate-transmitted diseases and injurious pests. So this can be anything from protecting people from West Nile virus, to the plague, to tick-borne illnesses, to dealing with cute little deer mice spreading hantavirus all up in people's cabins in the High Sierra. So just a little about our organization. We focus on disease investigation, surveillance and control, as well as education. Looking specifically kind of in order of importance, mosquito-borne diseases, tick-borne diseases, hantavirus, and then the plague, plus a smattering of other things that you can get from all of the biting arthropods out there. We have 3 field offices, one in the coastal region, which is highlighted with that blue star, which is where I'm located. This is also where our state public health laboratory is located. An office in Ontario down in the south that covers the southern half of the state. And then we have our Elk Grove office that covers the Sierra foothills and northern part of California, as well as our headquarters office in Sacramento, where CDPH is based. We are a small branch, a small section. We're nestled within the infectious diseases branch of a very, very large health department. The coastal region does cover quite a bit of area. You can see we cover from up in Mendocino County all the way down to Fresno County, which can really touch into some of those Sierra foothills as well. So Cal Public Health has been a PacVec partner for ages, and we do have many collaborations with researchers around the state, which lead to publications, special projects, serving on committees for graduate students. And as mentioned, our coastal region office is located at the state laboratory campus in Richmond, where we also have collaborations with our viral and rickettsial disease laboratory at the state, as well as our microbial disease laboratory at the state. So groups doing human testing, where we focus more on the environmental side of things. So early on this year, in 2024, we were contacted by Dr. Andrea Sway from San Francisco State University in San Francisco. She has worked with us on several tick projects. Her lab focuses on tick ecology in the coastal region. So given our proximity, as well as kind of our main focus in the coastal region, where we have a lot of tick-borne illness, a lot of tick-borne disease surveillance, she asked us if she could get an undergraduate training grant, would we be able to host an intern this summer? She had a student in mind. She was a rising intern. She'd been working in the lab. She'd done a little bit of tick collection, some tick identification, and she had some experience in running gels and PCR and doing several molecular techniques. So we actually thought that it would be a very good fit for us, because we do a lot of combination work between our environmental surveillance and our laboratory section. Anywho, so once we were approached, we said, give us a second. We need to think about how this would look, what we might need. So conveniently, last summer, we started an investigation of a human case of what we assumed was Rocky Mountain spotted fever. So we approached it the way we would do any environmental investigation following a tick-borne disease illness. We went out once we had identified exposure sites. So this case patient, who was quite ill, had exposure at golf courses in the Bay Area. The interesting situation here was his onset was quite late in the summer. So typically the dermacenter species that transmit Rocky Mountain spotted fever are sort of on the decline. They peak in early summer, but we went, okay, well, we're still going to get out there to the golf courses in these two counties in Alameda and Contra Costa. We worked with their local vector control agencies to go do tick surveillance at these areas. And we went out and we flagged all of the golf courses. And here's what we found. So at the end of July, early August, we went out and we collected dermacenter species ticks at all of the golf courses. Contra Costa number 2 had a surprisingly high number of dermacenter occidentalis found, especially for this time of the year, and not necessarily in an area where we were expecting to see them. So we did determine from a public health standpoint, because our goal is to protect public health and perform this generalized public health surveillance, ticks were present. There was risk of encountering ticks at these golf courses. All of the ticks that we collected were dermacenter species at this time, which means they're the classic vectors for spotted fever group rickettsiosis. However, all ticks tested negative for spotted fever group rickettsia at our viral and rickettsial disease laboratory. We also found that golf course number 2 in Contra Costa had the highest numbers of ticks. And golfers could easily come into contact with the areas that we flagged. So if you were, say, not a great golfer. I'm not a great golfer. I know I would certainly be out searching for a golf ball in these areas. I could easily come into contact with these ticks. So that was a sort of interesting risk factor for tick-borne illness that we hadn't necessarily thought about for this general activity. And it sort of led us to our future investigation goals, which were we should revisit the sites during the height of tick season when exposure likely occurred, since onset of disease versus when we actually get out there is often quite different. Post-recovery, we also found another golf course. We visited it as well, found ticks, not surprising. But this also led us to potentially distribute tick prevention materials at golf courses and also talk about risk with golfers and or groundskeepers. But this also, while all this is going on, our viral and rickettsial disease laboratory was also working on their own thing and sequencing these results. And what they found was it wasn't Rocky Mountain spotted fever. So that's interesting. And what they did find was this rickettsia aligned perfectly with a novel rickettsia called California's rickettsia subspecies 26, rickettsia lanei. We'll call it that for right now because the numbers just keep backing up in my head. But anywho, this pathogen, or now known pathogen, but previously unknown, was found to be found in Haemaphysalis leporus palustris, which is also known as the rabbit tick. These ticks traditionally are very host specific. They do not really get off the host. They spend all their time really feeding on rabbits in their environment. So the idea that this gentleman was infected by a rabbit tick, we thought was fairly interesting. It's not impossible. People do find nymphs on them at times, but given it was the spotted fever group rickettsia, they tend to be fairly rare in the environment. We wanted to basically start a bunny hunt and look for rabbit ticks and see if we could really figure out the incidence, or rather the prevalence, incidence and prevalence that we're seeing in these ticks. So we had a project. And after talking with Dr. Sway, we proposed that her intern come work with us, collect ticks around the San Francisco Bay Area where our case had been exposed, as well as where we found out doing some retroactive testing that there had been one other case with exposure out in San Mateo County as well. And we were going to look specifically for rabbit ticks and dermacenter species, which could be vectors as well. Just based on the frequency of biting, we figure we should really take a look, see if we can figure out if it might be found somewhere else. So investigating, we're going to test both the rabbit ticks and the dermacenter species for California 6269. And then also just sort of identify potential environmentally advantageous areas for rabbit ticks where we might be able to collect them more easily. Usually in our general surveillance, standard surveillance, doing our jobs on the day-to-day collecting ticks, we get about 15 of these ticks a year, all nymphs or larvae. And then with this, we hope to get a better grasp of the seasonality of these ticks in the Bay Area based on the fact that the case patient was infected in early summer and the general progression of this disease. Long story short, the training grant was obtained. Our intern started in June, ran through the end of August. Our results are pending if you'd like to learn more about them. If you're in California, you might consider attending the Mosquito and Vector Control Association of California's annual meeting in Oakland in January. She will be presenting a poster, or you can wait until the PacVec annual meeting where she'll be visiting and hopefully presenting on her results as well. So what did it look like for her as she was working with us this summer? An average week had about 2 days in the field. We would do tick collections at sites where rabbit ticks had been found in the past or exposure sites. And then that would be flagging, putting all the ticks in vials, bringing them back, looking at them under the microscope, because we're really targeting the nymphal rabbit ticks, but also we were getting all these other dermacenter species. And then she spent about 3 days in the lab doing tick identification. She, I would say, has moved up to pro level for identifying nymphal ticks. DNA extraction. So we collaborate with our viral and rickettsial disease laboratory for testing. So we do all of the extractions in our lab and plate them then for PCR for work in the, or for running the PCR in our viral and rickettsial lab. So it is great to have an intern. So just the benefits for us. First of all, we had more hands in a small laboratory. Our general laboratory staff is 2. So we have 2 microbiologists working on all of our different diseases at any given time. So that could be testing fleas for plague, doing chicken testing for West Nile virus, or doing tick testing as we do run quantitative PCR in our lab as well. We increased the numbers of ticks collected and extracted for spotted fever group rickettsia testing in general. We actually managed to collect close to 1,000 rabbit ticks this summer. So we greatly increased our numbers specifically with this species of tick. It gave us the ability to investigate a new pathogen, newly sort of found to be a human pathogen, on top of our general surveillance. So it allowed us to continue our normal activities while enhancing a very specific project that was identified through public health surveillance. And it continues to strengthen our relationships between PacVec partners, because it's great to have these connections with universities, especially when they have students who are willing to learn and participate and who are excited about doing science and public health in a way that may not be always highlighted in the media, since obviously, I won't say no one knew what public health workers did before COVID, but now you're getting a broader view of what it is. It's not necessarily just contact tracing. It can be outdoor environmental work, spending that time hiking it out and finding these, I think, wonderful critters, but others have different viewpoints on that. And it provides benefits for the intern. They get real-world public health experience in a large public health department. So they get to sort of learn what it really looks like when you're out there working within the constraints of one of these large organizations. Help provide training in tick identification, lots of experience in field surveillance, and training in molecular methods at some state-of-the-art laboratories that are just wonderful. But seeing how the different laboratories work, working with qPCR, and getting just a general overview of what it could look like if they want to pursue a degree or a future in public health as well. This will be leading to publications. We're continuing this on, continuing to collect rabbit ticks now that the intern is gone, so that we can have a better view of phenology as well. So it's some parts of the project are ongoing, and our intern will be involved with this moving forward as we move it towards publication. So with that, it is great to have an intern. Sometimes you have a mad scientist, as you have in Chris's ability, and sometimes you have a very structured sort of goal to work with. But I think either way you do it, you end up with benefits for both organizations. And with that, I am happy to answer any questions.
Thank you so much, Megan. That was really excellent. And as mentioned, your agency at CDPH and Salt Lake City MAD are very good representatives of the wonderful collaborations that we've been able to have through PacVec. And one question I have is, and one we've been struggling with a little bit with the PacVec leadership team is how to broaden these opportunities to more colleges and universities. And we have people like Neil Vickers in Utah and like Andrea Sway in the Bay Area who are established PacVec investigators. They have wonderful labs and funding from PacVec. And we've been wondering this year about how to offer these opportunities essentially to any 2 or 4 year college near you or that makes sense to you in terms of collaboration. And one of the tricks there is recruitment and also finding a mentor on campus who can provide a quality experience after the summer, perhaps. And I'm curious, let's start with Megan, maybe just to say what you would see as kind of an optimal arrangement or how you would find such mentors who are willing to maybe enhance the student experience once they're back on campus in the academic year.
That is a great question. I mean, from a PacVec perspective, I think we just need to remind people that there are a lot of great opportunities that can come with working with PacVec, certainly mosquito control in the state. I know my big thing was I wanted our intern to hopefully present her work in a setting where at like MVCAC at local conferences, because that's where a lot of students get their first experience with vector control. These are regional, they can be smaller, but we also provide great opportunities like poster competitions. So if you have a small project, you know, we send things out via the PacVec newsletter and MVCAC, but I really think we should be pushing it to those interested students as well, because just getting out there and presenting for the first time can give a student confidence. And if mentors know that they have those opportunities to share the work that they're doing to a broader audience that could interest either hosts or other researchers in what they're doing and promote those collaborations. Work with the regional conferences, because as Chris said, you know, he's a toxicologist. I'm technically an environmental scientist and public health practitioner. There are a lot of groups out there that don't really think about these connections until you have an opportunity. Like Chris's engineering situations, it's vector control brings a lot of connections and brings in lots of different types. So really pushing it out there and reminding our PacVec partners as well that maybe at their university, there may be potential collaborators that just haven't thought about fighting arthropods in general. So, you know.
Yeah. Chris, do you have thoughts on kind of the after internship experience, so to speak, and sort of how important that is to the total experience?
Yeah, so I know one of the things with recruitment on our side, and I don't know how this reflects across other members of PacVec, but in our case, in Utah, there's like a state requirement to use a platform called Slack, and or Handshake. I'm sorry, not Slack, but it's the same kind of thing. It's like a independent server to share information and things. And so like all the schools, like whether they're R03s or R01s, will disseminate announcements for things. And in fact, like us as a independent government operator, we have to put announcements on there, or we could be like in violation of like some of those communication requirements. So that for us is actually a hurdle. So it's like you have to make sure you like do this right and put it on the right mediums. But on the other hand of that, it means that the state, in our case, provides some sort of network to get information out to people and help with that recruitment. But you do bring up a good point about like, okay, after us, what do they do? I deal a lot with people in a very wide range of that like decision making. So freshmen aren't going to be looking for the same sorts of opportunities as a bachelor's who's considering grad school, as a master's, as a postdoc, and things. And so across that spectrum, my early professional, so like my first and second year students, I find that it's more important for them just to know that options are out there. A lot of them just don't realize that there are labs that they can ask. So after spending some time with us, I very seldom have them go seek out like a lab in particular. but rather they start to float around the department and start becoming part of the conversation, and they become like known names within their colleges and stuff, which it's kind of interesting. It confers its own benefits. Like they become like these floaters in a way. Whereas like my seniors and stuff that are looking for potential grad schools and stuff, usually what they end up doing is changing their focus about subdisciplines and things, and then looking for somebody that falls within our field. And so that's actually kind of where Neil falls in with us, is that when people start realizing, I want to work in this particular aspect of biology because of my experiences, then Neil can be one of those people that can suit that need. But that kind of comes down to the individual's ability or willingness to be part of it. So like Neil knows us, but not everybody at these campuses is equally like on friendly terms. And it doesn't mean they dislike us, but just that they don't know who we are, and they don't necessarily understand where the crossovers are and stuff. I will say that beyond that, when you get into graduate education and stuff, probably they've already found a lab, and what we're doing as the intern program is borrowing them. So that's kind of like the situation with Megan, where that student already was with Dr. Swipe, but you get to kind of get her on loan of sorts. And so that ends up being our case as well. So like Emily Calhoun, who's been supported through PacVec, is one of those cases where she has a major advisor. She has a lab looking at vector ecology, but we get to borrow her from time to time using stuff like PacVec. So it's kind of a spectrum with my side of it. And I think the only one where I think placement becomes an issue is really the people that have not yet decided where they want to land. So I think on the PacVec side, one of the decisions is it super important to you to help them find that place to land? Or do you just want to allow them enough means of communication to figure it out? Which is what I do. I point them towards stuff and just like, go talk to these people. I'm sure you can find somebody. And I'll do things like rec letters and like establish connections and stuff if I have to. But for the most part, it's them doing that legwork. I don't know how else you're going to do it, because I don't know if you have enough like kind of officially designated people within PacVec to just be like, oh yeah, we have like 15 potential advisors in Utah. No, you just have Neil. So, right. That's my suggestion on it. But that's kind of within the framework of how Utah is. And like, we only really have a couple schools that are trying to be active. We've got BYU, USU, and U of U. And like, so that's 3. That's very few for me to have to juggle. It's not at all like how it's going to be like in California, for example. So take it for what you will.
Yeah, I mean, that is what we're trying to do, is keep the core and the strengths we have. And we certainly have the investigators like Neil and Andrea who know you and know the work, and often are very happy to participate in these things, as would I be. But I think growing that spectrum to include potentially community colleges, potentially other biology instructors at colleges, even 4-year institutions, who aren't always vector specialists, but might be great mentors and might even get excited about this world of vector-borne diseases in ways they haven't to date. So that's what we're hoping to open up this year, is a little more broadening to keep what we have and grow that a bit in terms of reach and diversity.
You might be able to kind of pyramid it. So one of the things that we've been doing in association, not just with like PacVec, but also RAPVec, is just trying to find our own connections for stuff. So exactly. Our education specialist, Dr. Michelle Rabine, has been making really good inroads with a lot of these other colleges that we have. And so like, these aren't my personal connections, but they're people that invite her to teach and guest lecture and things like that. And so then we end up on this first name basis with some of these people. So one of the options could be, maybe it's not time efficient for PacVec explicitly to go make those inroads, but maybe you can make a key connection with like us, for example, that can help like share, that infrastructure that already exists, just because we're already building some of the culture. That's an option. I mean, of course, you already have a relationship with us, but I'm just saying, like, maybe you can do that across the network.
Yeah, that's kind of what we're thinking, is have partners like you, both of you really, and others propose internship opportunities in a way that once we sort of select the year's plan, you could go forth and recruit from your own resources, your own connections, which would potentially reach people that we wouldn't know about, as you're pointing out.
Yeah,
and like opportunities for others to ask questions here. Sorry to interrupt. I just want to make sure if anyone does have a question, feel free to unmute or put something in the chat. And Chris, sorry to interrupt. Go for it.
I was just going to say, like, social media sharing is pretty helpful. I do that with a lot of our campuses and stuff here, where we just like parrot each other's stuff. Um, so that's a thing.
Okay. Any other questions? Comments, things we can do better for internships? One thing we found useful is senior practicum projects, like any programs at universities that require a written project as a deliverable can be really useful for getting summer experience, coming back to campus, and ideally maybe your senior year or whatever that would be, you get opportunities to write something up. Even if it's not a publication per se, they get that scientific experience of trying to do the data analysis and craft the project in a written form.
I have a question for our attendees. Anybody who feels like speaking up, what's keeping you from going out and trying to get an intern? Crickets.
Maybe nothing.
Yeah, maybe. Well, but
And we have, I'll say, a number of people from CDC, CDPH, and Salt Lake City on the call. So I think, some of them drill well.
yeah, I mean, sometimes like all the infrastructure is in place, but you just don't have the time or the bandwidth. It's like I can't think about this right now. Like I can't manage anybody. Maybe you have the time, but you don't have the infrastructure. It's like money's not the problem, but the red tape is. You know, Everybody sort of has a different thing. But maybe I don't know if PacVec has the ability to kind of pull their user base for stuff, but maybe you could find out what seems to be common, and maybe there's some way to appeal to it somehow. Help people navigate that.
Okay, well, we will be announcing this opportunity for 2025 internships in the next week or so. So that really is aimed at you as partners, which will be the starting point. And then from there, we can figure out the recruitment angle. We do have a new mechanism this year where we don't have to only work with the funded investigators, which of course we're happy to work with all the funded investigators too, but we want to expand that to where essentially we could hire students anywhere in the region. So for any of you on the call, if you do have ideas for your local university or college, we would love to hear about that so we can form those as they make sense. So watch for that coming in the PacVec newsletter. I also want to mention in 2 weeks from today, which I believe is November 4th, we have Dr. Brian Foy from Colorado State University, who will be telling us about his work, and it's some collaboration with us at UC Davis as well on use of ivermectin for West Nile virus control. So he will be the speaker then, and we've got two more seminars coming later in November and early December as well in the same seminar series this fall. And with that, I just want to thank you all for joining, and especially thank you to Dr. Bibbs and Dr. Saunders for agreeing to present today. And we'll look forward to seeing all of you in 2 weeks. Enjoy your afternoon.
Thank you.